Good morning. Thank you so much for the introduction, Tanya. It's a great pleasure and a great honor to open this truly exciting symposium or workshop. Maybe workshop is even a better word because I really think of this as a working meeting. So for the next 45 minutes or so, I will talk about the expansion of the virus world. I changed the title the tiniest bit to use metaviromics. I thought that it was not a bad term, and metagenomic study of viruses has come of age to such an extent that I think we deserve this. We can treat ourselves with that. In this lecture, I will touch very little on the methodologies of metagenomic analysis. I'm sure you will hear a whole lot about this over the next 2 days. What I'm trying to do is really to convince everyone who still needs to be convinced that metagenomic analysis does not simply expand the world of viruses as we know it. It transforms our ideas of how this world works and evolves. This is the principal message, I think. So maybe I should have said expansion and transformation. All right. There is no need to spend time on this slide. In this room, everyone knows that viruses are the most abundant and, by extension, the most important biological entities on this planet. Viruses and metagenomes. Again, no need to waste much time here, but I just want to indicate a couple of things. The main is that the amount of metagenomic sequences already substantially exceeds the size of the regular sequence databases. So nowadays, when we search GenBank, it is very clear, or other traditional database, it is very, very clear that we are dealing with the tip of the proverbial iceberg. Another important point is that oftentimes, unless one really aims to study viromes, viruses may be ignored in metagenomic studies for various technical reasons. And another issue that is another aspect of metaviromics that I want to mention here is that there are really two types of detecting viruses, of studying viruses in metagenomic sequences. One is to study viromes per se, virus-enriched fractions. The other is sort of fishing out viral genes from general metagenomic data, in particular detecting all kinds of proviruses. Certainly, the best results are obtained when the two approaches are combined. So, virus metagenomics is coming of age and getting recognized, and in this time flies. In this consensus statement article in Nature Reviews Microbiology with a large group of colleagues, several of whom are here, we presented the rationale for the necessity to recognize viral species and higher viral taxa solely from metagenomic, from appropriate metagenomic analysis, from appropriate analysis of complete or near complete viral genome sequences extracted from metagenomes. And to my utter astonishment, this has been recognized by ICTV immediately, which to me is a sign of new times, that indication that the world, that our world of virology at least has really moved on. There are very, very good reasons for that. And they're simple. and can be illustrated by this graph coming from that very paper published a year ago. The most common and abundant viruses apparently in any environment, as we shall see actually during this talk, but in particular in the ocean, come from metagenomics and do not have an unequivocally identified host. So very simply put, here is prevalence, here is abundance, and the colored ones have known identified hosts. The gray ones do not. You can very clearly see that the substantial majority, including the most abundant and the most prevalent, the most common viruses, are unknowns coming from metagenomics. Ignoring that part of the world is unthinkable, I believe. So, as I said, there will be very little said today about methodological specifics of metaviromics, but a few words I do want to say. And this is more or less summarized on this simple slide, which says that viruses are good objects and bad objects for any kind of comparative genomic effort, and that based on metagenomics in particular. They're good because their genomes are comparatively small and comparatively simply organized with sort of wall-to-wall coverage by protein coding genes, by and large. So in that respect, they're relatively easy to analyze. But the other side of the same coin is that viruses change very fast, as we all know, and therefore, I cannot emphasize this more. It is crucial. It's really a game-changing issue. Using the most powerful computational analytical methods that you can find or develop. All right. So this is by way of introduction, and now to the matter at hand. Expansion of the virus universe via metaviromics. Clearly, when we are saying about expansion of the universe, much like in cosmology, that means that we cover new regions, new domains of the sequence space. But that is, in a sense, the more trivial aspect of it. What I would like to emphasize today is that we are discovering, and not sporadically, but systematically, novel genome architectures, new expression strategies for viral genomes. New biology, perhaps revealed is strong, but definitely predicted, and novel trends in virus evolution. We shall try to talk about all of it. The way I want to organize this is I think, rather natural and is based on this famous classification of viruses by type of the genome and expression strategies that has been first developed by David Baltimore almost 50 years ago. So when we'll look at the genetic cycles of RNA viruses, we really, saying it in a slightly techy way, viruses are the biosphere's laboratory of genomic strategies. So positive strand RNA viruses, double strand RNA viruses, negative strand RNA viruses, retroelements, retroid viruses that use reverse transcription, single-strand DNA viruses, and finally double-strand DNA viruses, which are the only group that corresponds to the genomic strategy of cellular life forms. So effectively, viruses span the entire logical space of genome replication, of the modalities of genome replication and expression. And we shall go through these different Baltimore classes. So on this slide that you will see several times in slightly different forms during this talk, I represent these Baltimore classes more or less roughly proportional to their diversity, and with these funny shapes showing different classes and families of viruses. So to begin with, we shall talk about, in some respects, the simplest, smallest, most quote-unquote primordial genomes in this virus world, those of RNA viruses. And here, what we observe here is that metaviromics really, literally exploded known RNA virus diversity. There have been many studies to this effect. It's impossible to cite all, but I think in terms of sheer expense of the diversity covered, these publications by Eddie Holmes and his collaborators from China really stand out. What these researchers do is actually to study the so-called holobiome. So basically they take an insect or shrimp or some other thing and just crush it and generate the transcriptome and fish out by sensitive computational methods genomes of RNA viruses. I think the results of this are fairly spectacular. So very recently, so still unpublished and not even submitted material in the next few slides, my colleagues and I have, inspired by these metaviromic studies expanding the diversity, they sought to revisit and reconstruct in a new way the evolution of RNA viruses. And what I'm showing you here is the resulting phylogenetic tree derived by a rather complex procedure from over 5,000 of RNA-dependent RNA polymerases and reverse transcriptases. We shall see, which, by the way, let's emphasize this, represent the only universal gene of RNA viruses. You'll see some details of the tree on the next slide. What I want to emphasize here is simply visually the dominance of metaviromic sequences that are colored blue here. They represent at least 50% of the diversity of viruses included in this tree. So here is the same tree with some specifics. And what we see here is rather striking. I think it gives us somewhat more definitive and new picture of RNA virus evolution as a whole. I think the relationship between different Baltimore clusters within the RNA part of the viral universe have been quite unclear to this day, but through this increased diversity and the detailed analysis, they are getting clarified. And what we see here is the nested topology, what in evolutionary biology is called paraphily, namely double-stranded RNA viruses emerge from within positive strand viruses, and in turn, negative strand RNA viruses emerge from within double-stranded RNA viruses. Much similar to the way birds and mammals emerge from within different branches of reptiles. So, notably, I'll try to say this, Negarna viricota namely negative strand RNA viruses, clearly come up as monophyletic, as a clade in this tree. And remarkably, this is the first viral phylum that will be recognized by that is effectively recognized by ICTV, just needs the final approval within a couple of months. And I suspect this is only the beginning. At least 4 additional phyla can be expected to come. This is about the sort of vertical course of evolution of RNA viruses. But I think, or rather we find, that the advances of metaviromics require us to reconsider also the modes in which RNA viruses evolved. In particular, realizing that the phenomenon that I denote here, horizontal virus transfer, HVT, in an obvious analogy to horizontal gene transfer, movement of viruses between different, sometimes very different hosts, plays a central, previously vastly underappreciated role in global ecology of viruses and of this planet in general. Basically, if we speak, for instance, about the origins of plant and animal viruses, but this is a general logical dissection of different evolutionary scenarios. So, which of course are not mutually exclusive, which all are likely to contribute to the actual evolutionary process. So it's either ancient common ancestry, or rather I should say, horizontal virus transfer, and or parallel origin of viruses with similar genomic organizations. So I believe until very recently it has been primarily accepted that the key aspect of the evolution of these viruses is ancient common ancestry, with some events of horizontal virus transfer and parallel origins. What we now see, however, quite consistently by analysis of different groups of these RNA viruses, and here I'm showing the schematic tree of positive strand RNA viruses of eukaryotes derived from RNA bacteriophage. It's a very interesting group of primarily mitochondrial viruses with the host ranges superimposed. Just visually looking at this slide, we can see that a very clear pattern. Extremely different hosts are associated with the same clade or on multiple occasions, suggesting, apart from any possibilities of contamination that still have to be analyzed carefully in each specific case, that horizontal virus transfer to a large extent has ruled the evolution. And the same kind of observation can be seen in the trees of other groups of viruses, in particular Picorna viralis, arguably, and their relatives, arguably the largest group of RNA viruses, which we believe now, based on many analyses that I cannot really cover now for the lack of time, is the likely founder group of eukaryotic RNA viruses. And this scenario that comes out of these analyses includes dissemination by horizontal viral transfer, which includes both plants and various kinds of animals, and which most likely played the key role in virus evolution during the terrestrialization of animals and plants some 400 million years ago. We see the same kind of patterns in the tree of mono nega viralis, an order of negative strand RNA viruses. And in general, I think this slide summarizes the conclusion that the current diversity of RNA viruses and the diversity of the host ranges and relationship between viruses infecting highly diverse hosts has been shaped to a very large extent by horizontal virus transfer, in particular horizontal transfer of viruses from reservoirs in invertebrate hosts to animals, to other animals and plants. Okay. So much of the RNA part of the virus world. Let's move to other parts and talk a little about the part that is largely responsible for that very first slide I have shown you in of my talk, which emphasizes the fact that viruses are the most abundant biological entities on Earth. Largely, or at least it's thought so, Cauda viralis, the tailed bacteriophages, are responsible for this enormous abundance of viruses. What I will try to show you is that even about with respect to this relatively well-characterized and enormously abundant group of viruses, metaviromics is a game changer. But a little bit about the current ideas on the evolution of large DNA viruses that have been developed in a sense prior to the current expansion of metaviromics. The greatest diversity of DNA viruses in terms of sizes, sequence space occupied, genome organizations, is actually seen in eukaryotes, and there appear to be two large domains of that eukaryotic double-stranded DNA viruses. One is represented by diverse viruses such as adenoviruses, pox viruses, and related giant viruses such as mimiviruses, about which we shall talk more. Extremely diverse group of double-stranded eukaryotic DNA viruses, of which the ancestral form seems to be represented by this group of palintoviruses, better known as palintons, namely self-synthesizing transposons integrated in many eukaryotic genomes that we now, however, know form viruses. And these are clearly related to relatively rare group of prokaryotic DNA viruses, primarily the tailless bacteriophages, such as TEC-T viruses. The Cauda viralis, the most abundant group of prokaryotic DNA viruses, gave rise to a smaller, even if also diverse, class of eukaryotic DNA viruses, which includes the herpes viralis. So here we have this little paradox that we will address, that common tailed phages gave rise to a relatively narrow group of eukaryotic DNA viruses. Apparently rare bacteriophages gave rise to a very diverse class of eukaryotic DNA viruses. Now we shall talk about this over the next few minutes, about this part of the DNA virus, of double-stranded DNA virus domain. So, it gives me a special pleasure to introduce this recent work on diverse family of bacteriophages that includes the most abundant viruses from our guts. So the story started with this paper coming from Rob Edwards' lab in San Diego State, in which they discovered, in a sense, an elephant in the cupboard. That is a bacteriophage that is by far the most abundant virus in our guts and actually in the entire human virome, but until this metagenomic analysis was performed, remained completely unknown, which immediately tells you something big about the state of our understanding of the virus world pre-metaviromics. But in a different sense, the results of this study were somewhat disappointing because the genome of the virus really looked like a desert. Very little could have been identified. So of course we could not leave it at that. And about a year ago, launched a reinvestigation of this cross-phage genome, relying heavily on metagenomic data. And on, as I emphasized in the beginning, on the most powerful available database searching methods, generating carefully curated sequence profiles for each of the proteins encoded by these viral genomes, and using these to recognize their provenance, identify their putative homologs. Fortunately, this was a rather successful exercise. And in particular, the key finding was that this virus is, after all, this cross-phage is, after all, not a complete unknown. If we try hard, we can confidently identify a typical caudoviralis capsid protein here, and a conserved gene block that apparently includes structural genes. Of course, we proceeded further and found interesting relationships among various proteins of these phages. In particular, confirming the affiliation of these viruses with Bacteroidetes, the most abundant bacteria in our guts. We find that at least some replication proteins of these viruses have been apparently derived from Bacteroidetes at an early stage of evolution. And there are other interesting features, such as unexpected links to eukaryotic viruses. I cannot really go through all the details here, but I do want to emphasize, in the spirit with which I started, discovering new biology through metaviromics, that what we predict in this new emerging cross-phage family of bacteriophages is a unique transcription apparatus which includes proteins of enormous size, combining multiple RNA polymerase subunits and proteases that are encoded either nearby or within the same protein, strongly suggesting that there is actually processing of these precursors to generate the mature RNA polymerase of these viruses. To make a rather long thing short, this genomic terra incognita has been charted as a result of these analysis by delineating the replication, transcription, and structural modules, and predicting the structures and functions of the majority of the proteins in each of these modules. Maybe I should have included an extra slide here just to emphasize that from this single bacteriophage genome that appeared to be complete orphan, we now can identify a novel family of bacteriophages that includes hundreds of diverse genomes. I want to proceed a little bit along the lines of Cauda-Virales, expanding this group now to a different domain of the microbial world, to Archaea, and in particular, Archaea. So this work that we performed together with colleagues from the Technion in Haifa, Israel, reports identification of a highly abundant group of viruses that apparently infect uncultured marine group Archaea, some of the most common microbes in the ocean. Although they are some of the most common, viruses of these organisms so far have been unknown. The story is roughly the same and involves identification of different modules in the genomes of these viruses and the signs of module shuffling, such as that the structural gene module is effectively of the bacteriophage origin, whereas the replication module, which is nearly complete in these viruses, they appear to be exceptionally autonomous in terms of their replication abilities, comes from Archaea. So there are a variety of phylogenetic connections, that the details of which I will skip. What is important to emphasize is that these viruses that we not very elegantly designated magroviruses after marine group 2 are among the most abundant ones, behind effectively only cyanophages and SAR11 phages in the viral fraction of ocean metagenomes. And when we perform a bipartite network dissection of the part of the universe occupied by Archaea viruses, we clearly place these viruses here with Cauda-Virales. So you have already seen this slide, but we will now talk about a different part of it, namely the apparent origin of the diversity of the enormous diversity of eukaryotic viruses, including the famous giant viruses, from a rare group of or relatively rare group of tailless bacteriophages such as tactiviruses. So we are now here talking about these viruses that replicate by protein-primed mechanisms that, however, gave rise to much larger viruses that replicate by RNA priming. So, very recently, in still unpublished work, we undertook, we made an attempt on comprehensive detection of viruses with double jelly roll major capsid protein in the prokaryotic world, thinking that perhaps, again, we see the tip of the iceberg. These tactiviruses perhaps appear rare simply because there are many others that we don't know how to recognize. So this is how they look, this is how they differ, two different major lineages of double-stranded DNA viruses, and so far there was very limited diversity of double jelly roll major capsid protein viruses reported for prokaryotes. This has changed, however. In this, in particular, in this major paper that happened to be published before we managed to publish our comprehensive analysis, but I think that's only to the better, reporting a putative new family, Autolyciviridae, that has a broad host range and is prominent actually in terms of being the major killer of bacteria in the ocean. So, very notably, the detailed search for double jelly roll major capsid protein encoding genomes uncovers a vast diversity of putative new viruses. Here is the picture before, and here is the picture after. At least an order of magnitude increase of the diversity of viruses with this type of structure. This is a completely novel group. If we denote Odin Archaea group, because in some of them, in terms of habitats, share the environment with the interesting new Archaea or the Asgard super phylum, and probably actually infect those Archaea. But apart from that, it's a vast group of viruses with double jelly roll major capsid protein and very small genomes. Here is the diversity of the genomic architecture. Basically, they share little other, only a couple of genes in addition to the capsid protein, but I think sufficient to identify a new family. And here is a phylogenetic tree that I particularly wanted to emphasize, because this includes this group of Autolyciviridae identified by Martin Poth's lab. We see that in the entire PM2 group delineated in our analysis, this is but a small twig in the tree. And another small twig is the previously known family Corticoviridae. Okay. Now, they move from these relatively small, relatively simple, and clearly clearly underappreciated viruses with double jelly roll major capsid proteins to the giants of the virus world. And of course, I'm very specifically pleased to introduce this, because this is the work that was initiated and largely performed by Frederick Schultz and Tanja Woike, and we helped with the analysis and evolutionary interpretation. Here we ask a very fundamental evolutionary question. Do giant viruses originate from a fourth domain of cellular life? So, just a reminder, using partly the same slide of giant viruses, mimivirus means micro-mimicking virus, and here it's shown side by side with a small, but not the smallest bacterium. You probably acknowledge that they look highly similar, actually. And these viruses are also famous for being infected by much smaller viruses that are somewhat close to adenoviruses known as virophages. All right. So the whole fourth domain of life, brouhaha, came from this, originated in 2004. Then the mimivirus genome has been sequenced, and amino acid tRNA synthetases encoded by this virus, which was a bit of a sensation in itself. These essential translation enzymes have not been seen in viruses previously, but the main sensation was that it was inserted into the tree of life based on proteins of the translation apparatus, and seemed to form, at least could be interpreted, as a separate fourth domain of life, descendant perhaps of unknown types of cells. Well, it's fairly easy to think about falsification of the fourth domain hypothesis. If the giant viruses in phylogenetic trees fall within one of the three domains, this is not compatible with the fourth domain hypothesis. If they are outside, we do not really know, but at least the fourth domain hypothesis has a chance. Okay. Frederick Schultz and colleagues have assembled from metagenomes, first from this sewage plant near Vienna in Klosterneuburg, then from many other environments, the genomes of what looks like a novel group of giant viruses. And even on this slide, visually, you can see that these genomes are very different in size, but more importantly and dramatically, they're very different in gene repertoires. However, they do encode the universal genes or the large of the nucleocytoplasmic large DNA viruses, allowing us to construct the phylogeny of core genes and show that they represent a sister group to mimi viruses. And also on this tree, the results of maximum likelihood reconstruction of gene gain and loss are superimposed. And what we can see is the extremely dynamic character of the evolution of these genomes, even compared to genomes of other giant viruses. The key thing, however, comes here. Close-Neu viruses encode a nearly complete translation system. You can compare visually here the close-Neu viruses, basically the entire almost the entire set of amino acid RNA synthetases in the largest viral genome, one missing, and a very rich set of translation factors, very different within close-Neu viruses themselves, emphasizing the dynamic character of evolution, but dramatically enriched compared to mimi viruses. However, and this could potentially suggest speculation on the origin of cellular host. However, these viruses also provide the most material for objective analysis of virus evolution. And when we do this, we see that the phylogenies of viral genes encoding translation components indicate multiple eukaryotic origins, suggesting that the dynamic character of the evolution of these viruses allowed them to acquire all these genes piecemeal. And the show is going on. Just very recently, a virus has been reported that actually is even more complete in terms of the translation machinery than the close-Neu viruses, and this is a more close relatives of mimi viruses. So the dynamics of evolution rule among giant viruses. We still need to understand the biology about the inordinate fondness of the translation system components. All right, the last I need to close with, the last thing to report novelties in the last novelty in the viral universe. And we now go to this part, to the smallest eukaryotic double-stranded DNA virus, the smallest but extremely important, papillomaviruses and polyomaviruses that are thought to have a very different evolutionary origin, namely originate from these single-stranded DNA viruses replicating by the rolling circle mechanism. You will hear much about these during this meeting, if I understand correctly. And we will connect them to this different domain of the virus world. Very quickly, this is again unpublished work in preparation, and in metagenomic studies of metagenomic surveys, again, holobiont analysis of different fish, several mid-sized circular double-stranded DNA viruses have been identified that are likely to become founding members of a distinct virus family. What is remarkable about these viruses that they combine features that have not been thought to be compatible until now, namely, a polyoma-like replication module, the so-called huge protein known as large T antigen that is essential for the replication of these circular genomes, but the adenovirus-like structural module. They're, so to speak, hopeful monsters of the virus world. So I will conclude here. I tried to tell you that metagenomic discoveries is becoming rapidly, or has become rather, the principal approach to chart the Earth virome. Novel virus families are discovered in genomes and mostly metagenomes by sensitive state-of-the-art methods of sequence analysis and structure prediction. Such methods have become more important than ever. And I will just submit that if you discover a novel virus these days, search the metagenomic database, genomic of course, but primarily metagenomic databases, very carefully. You'll probably discover a diverse and exciting new family. Importantly, metagenomics is far from being a trivial pursuit. Or rather, we get major insights into the evolution of the virus world, global ecology, and molecular biology of viruses. And finally, I'm not afraid to make a provocative statement, namely that the discovery of new families and even phyla, orders of phyla of viruses through metaviromics necessitate a complete overhaul of virus taxonomy, and I think we will live to see it. Many thanks to my collaborators, several people in the group who have been completely indispensable for this study, in particular Natalia Yutin, shown here, and Yuri Wolf, Mart Krupovich, and other colleagues from Institut Pasteur, with whom we have been working intensively over the last few years on evolution of different classes of viruses and theorizing on virus evolution. Valerian Dole from Oregon State University, my long-term co-conspirator in the study of virus evolution. Colleagues from Joint Genome Institute, Frederick, Tanya, Nikos, and others. I cannot put all the names on the slide, regrettably. Colleagues from Technion Haifa, San Diego State University, CDC, and National Cancer Institute. Thank you all very much for your attention.